Hi, I'm Kara Hoover, and I'm thrilled to be reigniting my 2018 Sci-Fu Lightning talk on building ancient noses. Since then, my team and I have analyzed 3 additional Neanderthal genomes, which makes a total of 4, as well as our Denisovan. We've also looked at ancient human genomes and several living hunter-gatherers and small-scale farmers. The question I wanted to answer was whether humans have a unique olfactory repertoire or whether we share one with other, albeit extinct, members of our genus Homo. I hypothesized that we would share a repertoire given that Neanderthals and Denisovan left Africa like humans and adapted to Eurasian environments, but starting 500,000 years earlier than us. As it turns out, I was wrong. First, the genetics. Our olfactory receptor genes bind odors, which enable us to detect elements of the olfactory landscape. In these graphs, receptor genes are on the y-axis, populations are on the x-axis, starting with ancient genomes on the left and then cycling through 1,000 genomes populations grouped by region. The left graph depicts variation in the gene, and the right depicts variation in translated proteins. The scale is relative to the human reference, so purple is no divergence, yellow is high divergence. The old noses have very little variation, as hopefully you can see in these distinct purple blocks on the left of each graph. Most of the variants they have are found in humans also. As much as 75% are shared in Denisova and 90% in Neanderthal. Gene fixation might explain this pattern due to these populations being small and isolated, but I would not expect their genes to be identical to modern human genes if this were the case. Rather, the pattern suggests to me that receptors are highly conserved, just like we see in chimpanzees and mammals more broadly. Our human pattern of high variation likely occurred after the split with the extinct members of our genus Homo. And this, of course, has reflected this gene conservation in the phylogenetic reconstruction from olfactory receptor gene data and why old noses form their own evolutionary clade in this orange box. Before we close the door, however, I did find a few variants that are novel in these extinct lineages. We used gene expression assays to test what odors activated each receptor, in which we found a novel variant. A few results are depicted in these dose response bar charts. Here, only significant results are starred, and these are when they differ from the control. The bigger pattern we noticed was that extinct and mysterious Siberian Denisovans were very sensitive to spicy smells. I've shown the response to honey on the left. The data suggests that their receptor is more highly tuned to honey than humans, who are equally tuned to honey and the caraway-smelling enantiomer of carvone. Denisovans were also sensitive to sweet smells, which is shown on the right. Were they keeping bees, getting takeaway curries from Tibet, and secretly snacking on cakes? Who knows? Neanderthals, another fun fact here, is that they were more sensitive to a variant of androstenone that's found in human male secretions like sweat, saliva, and semen. So meaty animal odors. In fact, that was the only odor this gene responded to, as indicated by the single starred bar in the dose response graph. You think the long cold winters of glacial Europe would reduce sensitivity to odors in very close spaces, but who knows, maybe it aided kin recognition. In sum, our relatives are generally highly conserved with few novel variants. When they do diverge, the variants alter odor sensitivity, not specificity. The raw data are on the left from dose responses and the predicted margins from the linear model on the right. The handful of novel variants examined suggest Denisovan is 2 times as sensitive to odors as humans and Neanderthals 4 times less. I also want to share our extension pack to old noses, in which we learned that humans were eligible for a genetic smell upgrade in the early days of farming in the Danube. I can't reveal the gene or the protein variant yet, but it is an on-off switch for sensitivity to the smells of hoppy beer and blue cheese. This mutation is prevalent in us, but missing in extinct lineages and ancient hunter-gatherers. A final farming-related mutation found in olfactory binding proteins is my last bit of data to share. These are proteins that bring odors through the mucus of the nose from the olfactory cleft to the receptors. The variant mapped here in pink reduces sensitivity to odors. Outside of Africa, the pink bubble grows larger, meaning people have that variant at a higher frequency. The frequency of this variant in living African hunter-gatherers, as depicted in the bottom left bar plots, suggests that perhaps the shift to domestication has something to do with it, given the frequency that we see doubling in living farmers, as well as 5 times that in 1,000 genomes. So hunter-gatherers use their noses daily for foraging, farmers do not. It makes sense that in these smelly environments of farming communities, permanently detuning olfactory environmental sensitivity would be a good thing. So I don't have time to show it, but the data for ancient hunter-gatherer receptors align with humans, which suggests again that the split occurred after the Neanderthal-Denisovan split and probably arose with domestication of plants and animals, mainly aspects of our genome changed when we stopped foraging and started domesticating. So if you want to get in touch with me and learn more about it, I've provided some of my contact information here and LinkedIn, and I'm just on time.